Welcome to the third video of our class. In the previous classes, we have dealt with the introduction to microbiology and we have classified them in general and also have seen a history of microbiology and applications. The second video, we have dealt with the prokaryotes and eukaryotes. We were able to differentiate between them and we have also seen similar components or cell organelles present in them and their functions. So today's class is different from those, which is a continuous of the prokaryotes and not eukaryotes. So what are prokaryotes? The best example is the bacteria itself. So today we'll be dealing with bacteria. In order to study, we first need to know why is it important for us to study about bacteria. Bacteria is a biological organism, isn't it? And these biological organisms contribute a lot in the nature. For example, 70% of the bacteria are helpful to the nature, that is eco-friendly, while the 30% are pathogenic and cause various diseases. Looking on its contribution to the nature, it is must for us to learn about the bacteria, and looking at its pathogenicity against the host cell is uttermost important to understand the bacteria and its role in causing various diseases, isn't it? So today's objectives will be first to identify the structure of a bacteria, understand what bacteria is, identify the various parts within the bacterial cell, and then we need to classify the bacteria based on various concepts, that is based on its nature, or based on the oxygen requirements, based on its morphology, that is the appearance, and based on its anatomy. Okay, so we'll be dealing all these in the coming slides. In order to understand bacteria, you first need to remember 3 points. The first one is bacterial cell contains a fluid-like substance within it. Every cell contains a fluid within it, isn't it? That fluid, we can call it as a plasma if it is a blood, right? Or if we consider a cell, there is certain fluid within it. If it is within the nucleus, it will be called as nucleoplasm, and if it is within the cell, it is called as cytoplasm. So that cytoplasm of the bacterial cell is also called as matrix. That matrix holds all the cell organelles, and it also contains various nutrients, waste material, gases, and enzymes responsible for the activity. The next point to remember is the cell envelope encases the cytoplasm and its components. What does it mean? What does encasement mean? For example, if we take a walnut, we have a shell outer side and the edible part is inner side. It means the outer shell is encasing the walnut with inside it, isn't it? Similarly, the cell wall or cell envelope that the bacteria has, it encases all the cell components within it. Not only bacteria, every cell in, let it be animal or a plant cell, contains a cell wall which encases all the cell organs. Okay. And the third point you need to remember is, unlike the eukaryotic cells, bacteria do not have membrane-bound nucleus. It means animal cell or plant cell, they contain a nucleus. Inside that nucleus is the nucleolus, which contains the genetic material. That genetic material could be DNA or RNA that condenses to form chromosomes, right? And that is responsible to give directions to synthesize various proteins, right? Similarly, in bacteria, there won't be any enclosure to the nucleus, but the genetic material will be placed randomly within the cytoplasm. You can see the structure of a bacteria here. So this is the cell wall. The blue color one is the cell capsule made of glycocalyx. Okay, every bacterial cell has a rigid structure that protects the bacteria from lysis, and that is called glycocalyx or capsule. Then followed by that, we have a cell wall, and after that we have the cytoplasm inside it. Okay, this cytoplasm contains ribosomes and mesosomes. This is the intrusion into the cell structure. It is called mesosome. And we have also a plasma membrane enclosing the cytoplasm. In the cytoplasm, there is a round color structure, which is the genetic material. It is a DNA. DNA in a circular form is called as plasmid. And this is the nucleoid, that is DNA, which is chromatin DNA, we can also call it. And the extensions are called flagella, that helps in movement of bacteria. And there are certain appendages called as pili. These pili helps in anchor on the host cell. It helps in attachment to the host cell. Okay, let us see each and every uh, component and their function. Beginning with the capsule, as discussed just now, it helps in attachment of to surface, protection against phagocytic engulfment, occasionally killing or digestion, reverse of nutrients, or protection against desiccation. It means the capsule that contains the pili. Capsule contains a pili, right? That capsule, because of pili, it will be able to anchor the host cell. It also protects from phagocytosis. What is phagocytosis? It means engulfing by lysosome and breaking it. So this capsule prevents the lysosome to show its action, the lysis of a cell. For that reason, bacteria cause greater effect in our body. And next is it occasionally killing or digestion. Okay, reverse of nutrients and proteins against desiccation. Desiccation means removing out. Okay, and cell membrane. Moving on to the cell membrane, as you can see here, the cell wall or cell membrane. It is of two types. It means gram positive and gram negative. Gram positive has, let us see the function, and in the next slide we'll see how gram positive bacteria has a cell wall and how gram negative bacteria has. Gram positive prevents osmotic lysis of cell protoplasm and confers rigidity and shape on cells. The cell wall of gram positive is thicker one. The thicker the cell wall, more rigid will be the bacterial cell, isn't it? Similarly, gram negative peptidoglycan prevents osmotic lysis and confer rigidity and shape. Outer membrane is permeable to barrier. Let us consider this is one membrane and this is another membrane. These two are the outer membranes, okay? And in between will be a peptidoglycan layer, and this peptidoglycan layer provides rigidity, whereas this upper and lower layers helps in osmotic diffusion, osmosis or diffusion of various ions. Okay, next is the plasma membrane. The innermost covering is the plasma membrane. Permeability, it has a permeability barrier, transport of solutes, energy generation, location of numerous enzyme systems. Okay, and the ribosomes, as you know, ribosomes are involved in synthesizing of proteins. It takes the RNA and starts synthesizing various proteins. And next is the chromosome. The whole part, the ribbon thing is the chromosome. Okay, and chromosome means it is also a DNA, right? And a plasmid. Plasmids are the circular DNA are called plasmids, right? And flagella, which helps in swimming motion. This is a flagella. Flagella is not just one. In bacteria, it can be many. We'll also see the number of flagella present while classifying it. And pili or six pili, this helps in anchoring to the host cell, or it helps in mating with another bacteria to produce different generations of bacteria. So stabilizes mating bacteria during DNA transfer by conjugation and or attaching to the surface. This will be the functions of the cell components. Okay, and moving on to the cell membrane, as just discussed, gram negative bacteria has two cell membranes, up and below, whereas the middle one has the peptidoglycan layer. And you can see this, the X shape, the red color one is the porine. Okay, this porine is the pore, which helps in transfer of ions, and also various other solvents, not just solvents, the liquid material or anything, it helps in exchange from those pores. Okay, whereas in gram positive bacteria, the peptidoglycan layer is very thick and has only one cell wall, which is the inner cell wall. You can see the cytoplasmic membrane, this one, and inner cytoplasmic membrane and the outer cytoplasmic membrane. And whereas here there is only one inner cytoplasmic membrane, whereas it doesn't have any outer cytoplasmic membrane. But this peptidoglycan layer is very much. What is peptidoglycan layer? We'll see now. Basically, peptidoglycan layer has teichoic acid and lipoteichoic acid. These two are attached one after the other and forms multiple layers like this. And these layers are attached such that these layers are attached in a zigzag manner. This teichoic acid contains two things, that is N-acetyl glucosamine, also called as NAG, and N-acetyl muramic acid, NAM, NAG and NAM. So here NAG and NAM will be attached. If NAG is here, NAM will be here. Okay, NAG is here, NAM will be here. Again, NAG is here, NAM will be here. In that way, the peptidoglycan layer will be attached. This is the gram-positive cell. You can see the peptidoglycan layer, very thick, and the plasma membrane here. Whereas here, there is a plasma membrane here and a plasma membrane here, right? That is the outer membrane and inner membrane. Whereas the peptidoglycan layer is very thinner one here, right? And there is a space between the membrane layer and the peptidoglycan layer, that is called as periplasmic space. So this was the cell wall of a bacteria. Next, moving on to the classification. There are different classifications of bacteria. First will be based on morphology, that is based on shape. Next will be anatomical, that is based on the anatomy of bacteria, that is appearance of the bacteria based on various parts it has, like flagella, right, or pili, based on that. And next is the staining. As you have seen, various scientists who have developed staining techniques to identify bacteria. So there are two types basically, gram staining and acid fast staining. We'll also see based on that. And then pathogenicity, as discussed earlier, it could be pathogenic or non-pathogenic, right? So we'll also see based on pathogenicity, its classification, and based on relationship of host and organism. It means in what way the microorganisms are affecting the host cell, or it is helping the host cell. And next, environmental factors, that is based on temperature, oxygen, pH of the medium, or salt condition, atmospheric pressure. Beginning with the morphological classification. In morphological classification, there are four different types. First will be true bacteria. Next will be actinomycetes and spirochetes. Sorry, there are total 5. So we'll see beginning with the true bacteria. In true bacteria, there are further two types, that is cocci and bacillus. Cocci basically means spherical or oval. Okay. Cocci, whenever you come across the word cocci or coccus, it means spherical shape bacteria. So spherical shape or oval shape. So we'll see the different types of cocci bacteria. Monococcus, it means single oval shaped bacteria. You can see the image here of monococcus, right? There are all the single cells are present of bacteria, and examples are monococcus species. Diplococcus, di means two, right? Diplococcus, so it means pair of bacterial cells attached together. So you can see here Diplococcus, two cells are attached together, right? And the example is Streptococcus pneumoniae. And Staphylococcus, in Staphylococcus, it appears like a grapes. When we see a bunch of grapes attached to one single branch, how does it look? Similarly, all bacteria will be attached, bacterial cells will be attached one after the other, such that it looks a bunch of grapes. Okay, here is the image of Staphylococcus in grape-like clusters. Okay, and next, Streptococci. Streptococci means one after the other, the spherical shaped bacteria will be attached. Okay, in this way. it will be a linear shape, okay, and it could as that of earthworm. You can see earthworm just like a thread like, isn't it? Not, it will be fatter than thread, but it is a linear, isn't it? The same way all the bacterial cells, oval bacterial cells, will be attached one after the other to form a linear chain. And the example will be Streptococcus pyogenes, okay, and the tetrad. The meaning of tetrad means four, right? So in tetrad, four bacterial cells will be attached in this way, two above, two below, and the other side two above, two below. So it will be like this: two, 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 two. So you can see here uh, tetracoccus. It means four. We can see, since it is a two-dimensional view, we are able to see only two, whereas it also has two behind, right? So it is tetrad, and the example is Micrococcus species. And then Sarcina. Sarcina means in a three-dimensional view, uh, in two-dimensional we can only see just four, as you can see here, just four, whereas in three-dimensional view it will be n number of bacterial attaching behind to it. So that will be a Sarcina, and basically it has around 8 cells attached to each other, or multiplication of 8. It could be 8, 16, 32, and so on. So that will be Sarcina. Let us have a quick recap. Cocci means spherical or oval, and cocci are different types: monococci, diplococci, staphylococci, streptococci, tetrad, and Sarcina. Moving on to the other one, that is Bacillus. Bacillus means rod-shaped bacteria. Whenever you come across a name with Bacillus, like Lactobacillus, the Bacillus means rod-shaped bacteria. So we'll see different types of bacilli, and first is Diplobacilli. The word itself says di, means two. So two bacillus will be attached to each other. Here you can see this, in this case, two bacillus will be attached to each other. And Palisade bacillus, it will be a sequence of bacterial cells attaching one after the other in this way, okay, one after the other in adjacent way. Whereas in Diplobacillus it will be in this way, and in this case it will be in this way. And then Chinese letter form, I didn't find the structure of it. And Coccobacillus, it means it will be like a spore coming together like, okay. And comma shaped bacillus, I didn't find that shape, that one also. And spore forming bacillus are also present. It will be like a spoon, thinner at one end and a bulged in on other side. That will be a spore forming bacteria or spore shaped bacteria. Moving on to the other types, Actinomycetes. These are true bacteria but appear like fungi as it forms branches and is filamentous. As you see grass on the ground, grass will be linear, single, right? But all the grass, it overlaps on the other like this, in this way. For that reason, it looks like wires coming together, right? Similarly, you can see the image here, how it looks. This is a cultured Actinomycetes, and when observed under microscope, it looks like this. So this is Actinomycetes, and next is Spirochaetes. Spirochaetes are long, slender, non-branched, and have coils within them. So what does it mean? It is very long, like that of an earthworm. Earthworm is not a bacteria or a fungi, okay? It is an animal. But in bacteria also, they are very tiny, that they are very longer in length, and they have coils within them, and they are not branched. You can see the image here, how it is: long, slender, that means thin, non-branched, and have coils in this way. The next is Mycoplasmas. Mycoplasmas doesn't have a perfect cell or perfect shape, so they lack rigidity because they don't have peptidoglycan layer to give rigidity to them, and highly pleomorphic. And fifth type is Rickettsiella and Chlamydiae. These are very small and obligate parasites. Moving on, based on anatomical features. What is anatomy means? It means what extra things it has, such as capsule, or able to form spores, or structurally could resemble to a spore, and having lateral appendages such as flagella. We have hands, we have ears. These all are the lateral appendages that we have. and these are based on anatomy, right? We have nose, we have legs. All these are extra appendages that we have to the body. Similarly, for a bacteria, if it has extra appendages, that will be based on anatomical classification. So based on anatomical classification, first is capsule. Certain bacteria has capsule, whereas certain doesn't have that capsule. The earlier image that we have seen, it has a capsule within it, but the structure is different. And from the intermediate that we have studied, the TMV bacteriophage, it has uh, a hexagonal shape of capsid. You can see here TMV bacteriophage. This is the capsid which contains the DNA, right? So this is the capsid. Such capsid are present to some bacteria, and for some bacteria it is absent. For example, gram-positive bacteria has a peptidoglycan layer, whereas gram-negative doesn't have peptidoglycan layer, but not as thick as the gram-positive. Similarly, based on capsule, it could have a capsule or it could not have a capsule. So capsulated and non-capsulated. Encapsulated, the example is Streptococcus pneumoniae and TMV bacteriophage. On the other hand, non-capsulated will be Viridian streptococci. And spore forming. Spore forming means it will be able to form spores itself, right? And the examples for them are two types. It can form a spore and it cannot form a spore. So spore forming bacteria are Bacillus species. Okay, all bacteria, Bacillus species, Lactobacillus, Acidophilus, and all. And non-spore forming bacteria, that is Escherichia coli. And this is the image of spore forming bacteria. Next is flagella. Flagella. These are the structures or the appendages that helps in movement of the bacteria or swimming of a bacteria. If the bacterial cell has one single flagella, you can see here it is called monotrichous. Okay, mono means one, trichous means having a flagella. Monotrichous means one. Lophotrichous, it means bacterial cell having appendages on both sides. It could be two appendages on both sides or four appendages on both sides, depends. So having flagella on both sides is called Lophotrichous. Whereas amphitrichous, it means bacterial cell having one single flagella on either side. Okay, one single flagella on either side. It will be amphitrichous, and peritrichous. It could have n number of appendages or n number of flagella that helps in movement. And the other one is aflagellate, which means bacterial cell which doesn't have any flagella. And example are Shigella species. Okay. Next, based on staining techniques. Staining is a method to identify type of bacteria present. The peptidoglycan layer that is present, that contributes mainly in staining technique. If peptidoglycan layer is present on the outside, that means gram-positive. It has a very thick peptidoglycan layer. Upon adding some reagents, its color will be changing. That changing color determines which type of bacteria. In case of gram-negative, it doesn't have a thick peptidoglycan layer. Why? The peptidoglycan layer is covered by two outer membranes. One is inner membrane and the outer membrane. And that membranes prevent the reagent to interact with the peptidoglycan layer. Therefore, it doesn't show any color. So there are two types of staining. First is gram staining and acid-fast staining. This staining techniques will be dealing separately in the next video. Okay, just for the classification thing, we'll see here. In gram staining, there are two types: gram positive and gram negative. Gram positive, it is further divided to cocci and bacilli. So gram positive cocci, which means spherical shape. Example is Staphylococcus aureus. Gram positive rods, that is bacilli. Example is Clostridium species. In gram negative, again further two types: gram negative cocci, Neisseria gonorrhea, which causes gonorrhea, and gram negative rods, that is bacilli, and the example is E. coli. In acid-fast staining, two types: acid-fast bacilli and non-acid-fast bacilli. Examples for acid-fast bacilli are Mycobacterium tuberculosis, whereas for non-acid-fast bacilli is Staphylococcus aureus. Moving on, based on pathogenicity, as we have discussed earlier, the microorganisms can be of two types, either pathogenic or non-pathogenic. Similarly, bacteria are different types: pathogenic, non-pathogenic, and commensals. We'll see what they are. Non-pathogenic means doesn't cause any harm to the body, right? And the best example is lactobacillus species present in our curd. Those bacteria helps in digestion of the food, right? And you can see the image of lactobacilla. Bacilla is rod shaped, right? So all these are rod shaped. And whereas pathogenic means which causes harm to the body, right? Which causes disease in the body. And the best example can be Mycobacterium tuberculosis, which causes tuberculosis, and Vibrio cholera, that causes cholera. And other examples are many, such as Salmonella typhi, that causes typhoid, right? Bacillus anthracis, which causes anthracis in cattle. So you can see the image here. These are bacilli because it is a rod shaped, right? And when we have the infection in our lungs, especially in tuberculosis, the Mycobacterium tuberculosis, you can consider this as a Mycobacterium tuberculosis itself. Okay. And common cells. What are common cells? These are non-pathogenic microorganisms. It can be either helpful or non-helpful or cause harmful. How is it happening? Common cells are those which are symbiotic with the host organism, that doesn't cause any harm to the body. But when the immune system of the organism or immune system of the host is decreased, these organisms can cause diseases. Okay. The best example for the common cells can be E. coli, which is in our intestine. Based on the relation between host and organism, what does it mean? Suppose if I have E. coli within my intestine, that is intestinal flora, it helps in digestion, then it is helpful to me, right? Whereas the same E. coli which is causing cancer to me is harmful to me, right? And there are certain bacteria which cause decay. For example, let it be a bread. It undergoes decaying process in presence of fungi. When fungi attacks on it, it starts decaying. Similarly, an organism, you might have seen various animals either in movies or in cartoons. When it is on the land, when it is dead on the land, after some days we will be just able to see the bones. What is the reason for that? Microorganisms deposit on those animals and consume their nutrition, they consume their meat, which acts as nutritional medium, and starts decaying, and therefore the whole nutritional medium, that is the meat, will be gone. So you can see here, saprophytes organisms live on dead matter and plays vital role in decaying process. You can see the apple here. The microorganisms has completely eaten it such that it is completely rotten. And you can see this here, the skeleton of an animal which is buried inside. The organism present in the soil has completely consumed the nutritious food, that is the meat, from the animal, and therefore it is left with just the bones. Whereas the parasites are those which cause diseases to the humans, right? The best example we have taken is the Mycobacterium tuberculosis, which causes tuberculosis. And here, to understand here, I've taken this image, and there's infection in the lungs, and upon magnifying it, we can see the organism present. You can see many round, that is cocci, are attached to it together. It could be coccus, right? Since it is a round shape, it is coccus. What type of coccus it is? And that is this kind of, it could be either Staphylococcus or Sarcina or Tetracoccus. Not Tetracoccus, it could be Staphylococcus or Sarcina, because it has n number of cells attached to it, right? See here, n number of cells are attached like a bunch. It could be either Streptococcus or Sarcina. And common cells are those which are non-pathogenic, right? And the best example is E. coli in our intestine. Moving on further, based on environmental factor, how does environment favor microorganisms, or how does environment favor bacteria? If we all know that bacteria will be dead at particular temperature, higher temperature, isn't it? But it is not the case. There are certain bacteria that can grow in cold conditions. We put food in a refrigerator. Why do we put it? With a concept that in lower temperature, the bacterial metabolism gets stopped. That means it goes to the dormant state where it cannot undergo metabolism. As a result, the food will be fresh, right? But it is not the case. There are certain bacteria which can grow in lower temperature, which can grow in ambient temperature, and which can grow in higher temperature. Similarly, based on oxygen, it could grow in the absence of oxygen or presence of oxygen. Based on pH, it could be in acidic pH or basic pH, and also in salt concentration. Normally, the solution that contains salt, there won't be any bacteria, but that is not the case. There are certain bacteria that can also grow in salt solution, which is containing low salt solution, and also which doesn't contain salt solution. Let us see what they are. In dealing with the temperature, first one is cyclophiles. Cyclophiles. Okay, cyclophiles are those uh, which grow between 15 to 20 degrees of temperature. Okay, the example is Pseudomonas fluorescens, and mesophils, which means 20 to 40 degrees. It could be ambient temperature, right? The normal atmospheric temperature. And example is E. coli, Salmonella enteca. E. coli, it is in our body itself, and our body temperature is 36.6 degrees centigrade or 98.7 or 98.6 degree Fahrenheit, isn't it? So it is appropriate temperature for E. coli to grow. For that reason, we have E. coli in our intestine. The next is thermophiles, which means the organism that can grow between 50 to 60 degrees centigrade. And the example is Bacillus stereothermophilus. And based on oxygen dependent, aerobe, which means it needs oxygen at ambient temperature, it needs 21% of oxygen. Whereas obligate anaerobes are those which cannot survive in the absence of oxygen, and oxygen is required in huge concentration. Example are Pseudomonas aeruginosa and microaerophilic, which means these microorganisms are able to survive even if the oxygen percent is 5 to 10% and carbon dioxide temperature is higher than oxygen, that would be 8 to 12% or 8 to 10. So you can see microaerophilus are those which require 5 to 10% of oxygen and 8 to 10% of carbon dioxide. And the example is Helicobacter pylori. Based on pH, what is acidic pH? What is basic pH? The pH below 7.0, let it be 6.9, until 6.9 it is acidic, and from 7.1 to 14 it is basic. But neutral pH is 7.0. Let us consider 6.5 to 7.5 as a neutral one, since 6.5 to 7 is a very lightly acidic and 7 to 7.5 is very lightly basic, right? So we'll consider 6.5 to 7.5 as a neutral medium. Acidophiles, which means lives in the acidic condition, that is below 6.5 pH, whereas alkaliphiles are those which live in alkaline pH, that is above 7.5. Neutrophiles are those which survives in the pH of between 6 to 8, we can consider, but there are certain bacteria which are included in neutrophiles that can survive in 9. So let us consider from 6 to 9 as a neutral medium now, okay, just for the sake of bacterial survival. And next one, based on salt concentration, halophiles. Halophile, basic thing, halo means halogen, presence of halogen. Halophiles, halogen presence. What is halogen in a salt? That is NaCl, chlorine, right? Chlorine itself is a halogen. So halophiles, it means salt concentration. The organism which can survive in greater salty solutions, or greater the solutions having more concentration of salt, they are called halophiles. Example are Marinobacter lipolyticus. Marinobacter, you can by the term itself you can understand marine. The marine means marine water is very salty. The bacteria that grows in marine can be called as halophiles. Whereas non-halophiles are Enterobacter flavors. Non-halophiles are those which doesn't survive in presence of salt, right? And this was the introduction of the bacterial cell and the classification. So today's class we have studied the basic structure of a bacteria and the contents of a bacteria, and we have also classified the bacteria based on morphology, anatomy, staining methods, pathogenicity, relationship between the host cell and the organism, and environmental factors. So this was today's class, and these are the references. The best book to study microbiology is Pelzar, and followed by you can study B.D. Singh just to study about the cell and its components, and also animal biotechnology by B.D. Singh. So thank you for watching the video.